I'm going to talk today about just some recent advances that we've seen in vector biology and disease spread. And like Mark, I'd also like to thank you for your continued donations to AVF, for your assessment that you pay into the PD GWIS program, and also into the Napa Wine Grape Pest and Disease Control District, because I couldn't really support the team without that. So a little bit of start. Of course, we've been talking about red blotch and leaf roll all day, but I just want to emphasize that the ecology is really important, and that's where we focused a lot of our work, is really understanding what's going on in the system with the host, which is Vitis species. So red blotch is not found out of Vitaceae. We have tested, Mark and his team tested a lot of cover crops. We tested a bunch of the plants that we collected for the gut content analysis, and Vitaceae is the host of red blotch virus. So don't let anybody tell you it's in anything different. We have not found it yet. And as Mark talked about, Spicistolus festinus, the vector, has many breeding and feeding hosts, and so I'll talk about those today. So tika, yes, historically a curiosity. When I started working with leaf roll in 2009, one of the first things I always trained the scouts on is, if you see just a small cluster of red leaves, to always go look at that lateral, look at the petiole, make sure that's not girdled, because we don't want to mark that as disease when it's just a girdle. So tika is something that we've sort of followed the damage of, just because we don't want to confuse it with disease symptoms. And you know, when we actually started, like Jake, finding them on the petioles, it was kind of interesting and curious, because they are a little bit cute and they like to run around the petiole and hide from you. So it's kind of fun to see, move around and watch them, move around the petiole and hide from you. As we mentioned, there is another membracid or tree hopper in Napa vineyards. We find it similar to tika. We find it sort of north to south, east to west, up into the hills, down in the valley floor. There's some groups who are looking at it as a potential vector. One thing I will say is that there are certain aspects of its biology, namely when it occurs in the vineyard, that would suggest that even if it is capable of transmitting red blotch virus, whether it is epidemiologically important or relevant outside of small areas is questionable. So it's similar to, if you think about Aphrophora spittlebugs and Xylella, and that they're really only important as vectors in certain areas for PD. So torticollis may be the same. Again, it has not been confirmed that torticollis can transmit red blotch. And you can see we actually catch a lot of specimens in our traps. So my team has gotten pretty good at, distinguishing between torticollis and tika, but we've caught, over the 3 years, we caught 758. Of those, only 6% were carrying red blotch. So if that gives you an indication of the comparison between the number of infective tika that we find versus the number of infected torticollis, that goes back to my comment around how epidemiologically or ecologically important that species would be. So Mark showed you the photo or the map, thank you, Sarah, of how the captures of tika are distributed, again, north to south, east to west. The number of tika that we find in traps varies by year, as Mark talked about, and also by site. And so some sites we sort of consistently get a same number of tika year to year. Other sites we find them some years, not other years. And so there's a fair amount of variability when even places where we do find tika year to year, how many we're finding in our traps. Mark talked about infectivity, just a different way to look at infectivity here. Again, we had significantly more infective tika in 2021, because they were driven into the vineyards by the dryness of the other vegetation compared to 2022 and 2023. And then if you look at infectivity through a single growing season, we had significantly more testing positive for red blotch in August than we did in June or July, and very few infective specimens or even in specimens at all in May or October. So here's the gut content analysis again, and this is to emphasize where this insect is feeding. And so if we think about it, cultivate vinifera is actually an alternate host of tica, right? It's opportunistic. It's the fact that it exists in the landscape. And so at a certain period when a lot of the other plants are dried up, the insect will go into the vineyard to feed on vinifera. So let's distinguish feeding from breeding host. The breeding host work, a lot of that work has been done in the greenhouse, but we also know it historically from the crops in the southeast where tica is found. So when you think about breeding hosts, and this is important, those are mostly in the Fabaceae family, and principally those that are closely related to alfalfa, right? Alfalfa is the historical crop for this species, and so it makes sense that breeding would happen on clovers and vetches, which are closely related to alfalfa. We've never been able to confirm vinifera as a breeding host, but we did some trials in the vineyard to look at breeding hosts in the vineyard, and this is when we found that in our inoculation trials of Californica that we did have reproduction on Californica. So the adults lived for a long time in our cages, and then happily we did, well, happily for us, right, because we're researchers, but we did find some nymphs, and that's what you see crawling in that cage with the bird clover is one of the nymphs that we found. So the Asteraceae, like Mark said, they're very common in the gut. We did find that the tica lived for a very long time on bristly ox tongue. So 8 weeks later, we were surprised that that tica was still hanging out on that plant. But we did not find any reproduction in our cages on either the Asteraceae or the Plantaginaceae, but we did find that they reproduce on bird clover and Spanish clover. And so if you look at all the species that we found in the gut content analysis, here are some of the Fabaceae species that we found. And again, they're sort of closely, not surprisingly, some clovers, peas, and vetches. So where is bird clover? Well, if you're my dog, you can find it anywhere, right? But she finds it on the drive roads. She finds it when she goes into the ditch, when she's, searching for something fun to chase. So it is fairly common. As John Roncoroni would say, we weren't all that worried about it because it goes dormant somewhat early. It doesn't grow very tall. It's not super competitive. Oh, look, and it's fixing nitrogen. So I think that we've rationalized it as like, oh, it's an okay weed because it's doing that sort of thing. Well, it also happens that it is harboring populations of tica. So Spanish clover is a later season plant. So these photographs were taken in August and September, and just like bird clover, I find it in ditches. I see it along the trail. That's the Silverado Trail there, so I see it along the drive roads. And then also in some cases in vineyards. And it's perfectly happy in August and September, setting seed and flowering, and so it's sort of a late season flourisher. Vetch As you can see, in some years it grows pretty large, sometimes in the vineyards, it was in cover crop mixes, but then it's also escaped into adjacent grasslands and oak woodlands. And not surprisingly, in our study blocks, we see these patterns of disease spread near areas with weedy ground cover. So this is what Sarah was talking about in the panel, around reservoirs, along ditches, along drive roads. There are Jennifer and Hannah looking in that area here between these two blocks that have been neighborly sharing red blotch back and forth across the years. And we're there looking to see if we can find any tika nymphs. So we did look at a lot of burr clover in the spring and other potential hosts, and we did not find any, but that doesn't mean that they're not there. It just means that they're difficult to find. And so this is the part where I talk about the vegetation management and habitat manipulation that we've been thinking about doing for tika. So to date, a lot of that has really focused on these vineyard cultural practices. So what Jake was saying, based on these studies that came out, Cindy talking about the degree day model for when we should cultivate, if we wanted to interrupt the life cycle of tika on cover crops. And then also a study that came out of UC Berkeley looking at the difference in the trap captures between something that was clean cultivated versus mowed. However, as we've seen, this is not a vineyard specific insect, right? This is something that moves between our cultivated landscape and natural habitats. So focusing our efforts on cultivation in vineyards doesn't seem like it's really going to address the root issues here. Mark didn't show this, but the early season gut content analysis, how many cover crops do you see in that list of species? What do we see mostly? So this is a test of your trees. Yeah. So at the time of this year, when we're finding tika, these tika were caught in vineyards too, at the edge or in our traps. That we're not seeing species that are grown intentionally as cover crops in their gut content analysis before we do any sort of cultivation or mowing of that cover crop. Another way to look at it is we've taken the field sites that we've been working in, places where we've been following tika populations and disease ecology over the last 4 years, and looked at the landscape diversity around those sites. So we looked within a half a mile radius, and then we categorize the diversity of habitats around those into those different categories that you're seeing. So agriculture, and then natural areas like coniferous forests, oak woodlands, riparian woodlands, and shrublands. And then we took our trap capture data. So what you're seeing in the graph there, the ones that are above the y-axis, those habitats have a positive influence on tika occurrence in our traps. So we're more likely to catch tika when our study site is surrounded by grasslands or oak woodlands, and less likely when it's surrounded by vineyards or riparian woodlands. Okay. So Mark said, well, don't, just erase any other diseases from your mindset. I can't do that. It's too hard. It's all built up historically, right? So I start thinking about Pierce's disease and blue-green sharpshooters. And one of the principal things that we do for PD in the North Coast, where blue-green sharpshooter is our principal vector, is we reduce breeding hosts like tika, Blue-greens feed on a lot of different plant species. We're not eliminating all of their feeding hosts, we're targeting breeding hosts to reduce populations that are coming into the vineyards infected with Xylella and reducing our PD epidemic. So places where this has been done and where it's been done over a long period of time, such as the Rutherford Reach of the Napa River, the amount of PD and the PD pressure in that area has significantly reduced since they have done these practices. So I'm proposing that if we were to consider some way to think about habitat manipulation, that we would think about these persistent leguminous breeding hosts both in vineyards and in adjacent habitats to reduce three-cornered alfalfa populations. And again, breeding hosts, what did Mark say? Males are most important. Well, what do males do? Well, of course, they follow females because, who wouldn't? If you reduce the likelihood that the females are going to be there for breeding, then that is maybe reducing the likelihood that the males are going to follow them. So if there's no female population hanging out there on the breeding host, then perhaps we have fewer males as well. Okay. So you've been here all day, so you know that red blotch disease is concerning. If you haven't had personal experience of just how explosive it can be in certain areas, I'll show you this block where in 2022 we had 115 newly symptomatic vines, 2023 117, 2024 we had 768 newly symptomatic vines. So we do get very high rates of spread, and there are a lot of interannual differences, and again that goes back to all of these fluctuations in how many teak are in the environment in a given year, and then also how much feeding they're doing in the vineyard in that year. So we have been following the spatial patterns at these study blocks, and just like Mark was saying, we see these two patterns of spread. So we see these highly aggregated areas, but then we also see random spread. So in the highly aggregated area, again, you're getting mostly that sort of jumping behavior. Again, the common name tree hopper should indicate that hopping is a principal mode of dispersal as compared to the tree hopper flyer, right? Like, they're not doing a lot of super long distance dispersal. Obviously, they can do it because we do see these random patterns up to half a mile from where an aggregated hotspot is, but most of the patterns are in these aggregated patterns. Some blocks are lucky in that they only have the random spread. And that means that hot spot is located somewhere at the neighbors, not close enough to allow for that short distance travel, but close enough that we do still get some random hopping into that block. So the random spread, we seem to be able to successfully manage with vine-by-vine roguing. So you can maintain a fairly steady amount of interannual disease by going out, mapping at the right time of year, flagging those symptomatic vines, and removing them. Again, you need to optimize those to symptom development. If you haven't visited our table in the lobby or seen our publication, we wrote some best practices around monitoring for visual symptoms of red blotch that really go into some of the nuances of how the symptoms develop, both through time and then also on an individual vine. The issue that we've been having is that our vine-by-vine roguing efforts have not reduced spread in aggregated areas. So we keep removing all those symptomatic vines and going out, and instead of the epidemic going, downhill, it still continues to climb. And so that's where we really focused a lot of our efforts. Part of that has to do with the incubation period, right? So with leaf roll, we inoculated vines with infected or varioliferous mealybugs, and we saw that a year later we had symptoms. That was really important because then we knew that, okay, if we went out 1 year and removed vines, the next year, those vines that we were removing were symptomatic, were not newly infected, but they had been infected in the previous growing season. Well, unfortunately, as we've suspected with red blotch for a long time, there's an unusually long incubation period. So the time between when Tika infects that vine and when it shows symptoms could be up to 3 years. So this is observational evidence from both Mark's group and from some of the work that we've been doing, suggesting that we're looking at a 3-year period between when the vine becomes inoculated and when you start seeing symptoms. So what does that mean? So if you pulled out this year, you pulled out all of your symptomatic vines from 2024, well, those transmission events might have happened back in 2021. And so you still have in that block the infected vines from transmission events that happened 2022, 2023, and 2024. And so that's long enough that we cannot get ahead of that by just pulling out the symptomatic vines every year. We have to some way account for, in our roguing efforts, all of those vines that are asymptomatic but infected. And as Mark said, our zonal roguing strategy needs to account for those vines. So with our spatial roguing with leaf roll, we're pulling out the infected vine and 2 adjacent vines along the row. That can accelerate the rate at which we can reduce the disease spread for leaf roll. But because we have such a longer incubation period for red blotch, we need to think about that as zonal roguing as compared to spatial roguing. So Mark showed some suggestions for how spatial roguing might be, or zonal roguing might be approached based on the Cabernet Franc block that they've been working in. We are also working, because we've been studying 12 other blocks, and we want to compare the findings from that Cab Franc block to the places where we've been working. And one of the ways that we're looking at that is to look at how the newly symptomatic vines in any year and how close they are to previously known infections. And so most of the newly symptomatic vines we've found are within 20 to 30 feet of their previous infections. So that's starting to get between Mark's work and the stuff that we'll be doing with our data now that we have 4 years of data. We'll hopefully be able to give you some more specific guidelines around the parameters for zonal roguing. So the key take-homes are that if you're going to reduce spread in aggregated areas, you have to do zonal roguing. So you got to take out everything that is symptomatic and then at some distance beyond that. And you also need to eliminate any adjacent or neighboring hotspots, because I've seen it over and over again where one block gets pulled out, but the neighbor still has a large hotspot. It gets passed back and forth. And before you know it, a 6 or an 8-year-old block has so much red blotch that it's now being replanted a second time for red blotch. And I don't need to talk about it too much because you went through a whole collaboration session this morning with Malcolm and Carrie, but talking to your neighbors, building these coalitions and collaboration is going to be very important for managing the spread of red blotch. So one question we get, of course, is how can we actually just detect all of these? So we would just know which vines are infected but asymptomatic. And the answer is sort of. So we've been working with the LAMP assay, doing trunk cambium sampling, and here's what that looks like. So we had vines that in October 2021 and 2022 did not have any symptoms, and then in October and November of 2022, we did a lamp assay on 75 of those vines. And then the following growing season, we assessed all 75 of those vines for whether they had symptoms or not. 39 of them had symptoms, and 36 of them did not have symptoms. And so the chart that you're seeing is based on that. So on this side, you're seeing the vines that developed symptoms, and here the ones that did not develop symptoms. So of the ones that developed symptoms, with trunk cambium, we detected those infections with the lamp assay for 64% of those. 36%, they went on to develop symptoms, but we did not detect those infections. With the cane samples, we detected 28% and didn't detect 72%. With the petiole samples, we only detected 5% of those infections. Interestingly, in the symptoms absent column, we did have one vine that tested positive with cane samples, but did not have symptoms. So the answer to can we detect those asymptomatic vines is that yes, sort of, but not all of them and not with full confidence. So you're tired, it's been a long day. Thank you for your attention. Thank you for your support. I work in many of your vineyards, and we wouldn't be here without you. So I don't know if I have time for questions or if everybody just wants to go.
Anyone? Yes.
Two questions.
Yes.
First one, there got to be some natural enemies of Tika, like birds or insects. Is anybody working to identify those?
Yeah, so it's similar to blue-green sharpshooters in that these are not large populations of Tika, so we're probably already getting some biological control. Whether we're also going to then send maybe chickens or ducks out in flocks to try to eat them is a question.
Second one, any research on different rootstocks? Is there such thing as a rootstock that is less affected by red blotch?
There have been some studies on rootstocks, and there are rootstock differences in how the vines respond. There are varietal differences. There's also site differences. We don't fully understand all of that yet. And there's also variability within red blotch, so it's not a single genotype of the virus. So we don't understand how environment, rootstock, scion, viral genotype interact in a situation with, other management practices. So that gets really complicated, and you need a lot of sites because there's so many variables that you need to account for.
So with that rootstock question, you showed us that Vitis californica is or could be a breeding host. Do rootstocks kind of fall into this category of free-living vinifera? As in, can the Tika
reproduce on Riparia or Rupestris or
Right.
Not as far as I know. Where'd Mark go? Not as far as I know.
Thank you.
But I don't know that anybody's really tried with all of the different rootstock species.
All right. Thank you so much, Monica.
Sure.